All right, I think we should start now. Okay. Welcome everyone to today's Tech Talk. It's titled Immunopeptidomics, Phosphoproteomics and Olink, State-of-the-Art Technologies to Study Antigen Discovery, Cell Signaling, and Biomarker Discovery. This is brought to you by the Monash Partners Comprehensive Cancer Consortium, the MPCCC. My name is Tu Nguyen Dumont. I'm a cancer geneticist and bioinformatician in the Department of Medicine at Monash University. And I'm the convener for the MPCCC Tech Talk series. Before we start, I'd like to pay our respects to the traditional custodians of the lands from which we join together today and acknowledge their elders, past, present, and emerging, as well as any members of the Aboriginal and Torres Strait Islander community who may be among us this afternoon. To set the scene, the MPCCC Tech Talks aim to provide an informal and interactive forum for our professional research community to discover and discuss the latest cancer research technologies and how they can be applied in practical ways to support our research. The Tech Talks offer plenty of time for Q&As at the end, and we really do encourage you to tell us about your experience, key learnings, tips and advice, tips and tricks, as well as any potential challenges and limitations. And today I'm really pleased to welcome Dr. Pouya Faridi and Dr. Terry Lim as our guest experts. Pouya is the head of the Translational Antigen Discovery Lab at the School of Clinical Sciences. He's also the director of Clinical Proteomics Node at Monash Health, Monash University. Following the completion of his pharmacy doctorate and PhD at the Shiraz University of Medical Sciences in Iran, he joined Professor Rudi Abersold's laboratory at the Institute for Molecular Systems Biology at the ETH in Zurich, Switzerland. In 2016, he moved to Australia and joined Professor Tony Purcell's lab at BDI Institute at Monash University. And in 2021, he established his own lab at the Department of Medicine at Monash Uni. Our second guest expert is Dr. Terry Lim, who currently holds a joint research fellow position in Dr. Pouya Faridi's lab and the Monash Proteomics and Metabolomics facility. Terry graduated with a Bachelor of Biotechnology with first-class honors, specializing in medical biotechnology, and subsequently completed a PhD at Monash University under the supervision of Professor Tony Purcell. Terry's interests in cancer biology has led to a postdoctoral research fellowship in Professor Roger Daly's lab. So they're going to give us a presentation about 3 state-of-the-art technologies in the area of proteomics. They will explain how immunopeptidomics will enable us to discover targets for immunotherapy of cancer, autoimmune and infectious diseases. So during their presentation, could we please ask that everyone please mute your microphones and turn off your video to minimize disruptions, and we'll take questions at the end. So thank you very much, Pouya and Terry, for accepting our invitation today, and I'll hand over to you now.
Cool. Thank you very much for the invitation, and thanks for very nice introduction. I start this presentation, and then Terry will continue that. Let me share my screen. Hopefully you see my screen. All good? Okay. So I'm Pouya. As you said, I have some 2 roles. I'm head of Translational Antigen Discovery Lab at Monash University. Also at the same time, I'm a director of Clinical Proteomics Node. It's a new node which is part of the big umbrella of the proteomics facility at Monash University. Started last year. And clearly, what we do is proteomics. So the first question is why we need proteomics? So you know very well, definitely you heard about different omics technology. So we have genomics, which study genome and the process that it will study replication. Then we have transcriptomics, which is another level after genome. We study RNA and then transcription, and then proteomics, which study proteins, and it's like about translation. As we know, and we know very well, lots of things that are happening in the cell, signaling, and lots of functions that we see in our cells is because of proteins that we have. And we always wanted, scientific community always wanted to know what proteins are in the cells and what's their abundance, what's changing in different diseases. Okay, so this is like a very general workflow that we have in proteomics. We can have different kind of samples. It could be cell lines, it could be body fluids, it could be tissues, it could be from human or animals or any other kind of biological samples. What we usually do, we extract proteins, and then after extraction, we digest these proteins to small pieces called peptides, and then we run these peptides in a machine called LCMS, liquid chromatography mass spectrometry. So this is the heart of proteomics studies. And then when we got some signals or some ions, some spectra from the peptides that we sent through this machine, then we have different bioinformatics workflow to find what these signals or what this spectra means. So translate spectra to peptides that we have in the proteome. We usually have a template for all of this proteome. And what we do identification and quantification for these proteins. And then after we know what's changing in the proteome level, we can do lots of data mining and then understand what different pathways going up or down. So this is something that we can say, okay, so we can do a very similar thing on transcriptomics or RNA signal analysis. But what is very unique for proteomics is that we can also look at these post-translational modifications. So post-translational modifications that happens on the protein level on different amino acids that we have. There are tens of non-post-translational modifications, as you see the small list of that here, and then based on the different tissue, different diseases that we have, these post-translational modifications are different. So we have different technologies that we can enrich our samples using different techniques to particularly look at these post-translational modifications. So we don't have this kind of data on RNA-seq, and then we run samples on mass spectrometer and then do data analysis. And Terry will talk about one of the most well-known post-translational modifications, which is phosphorylation, and how we can do phosphoproteomics and what we can get from that type of analysis. What I want to talk more about this today is what we are doing on antigen processing and presentation pathway. So this is a pathway that tells the immune system what's going on in the cell, inside the cell. So all the endogenous antigens that we have in the cell when we don't need this protein anymore go through a process called protein degradation. The core protease here is proteasome. Proteasome cuts these endogenous antigens to small peptides, and then this peptide come and make a complex with HLA molecules, HLA class 1 molecule, and then this complex come to the cell surface. And then T cells, in particular CD8 positive T cells, monitor what's presenting by this complex on the cell surface, and if they find something is not usual, it means immune system is not trained with that. It find this complex and lyse itself. So we have another pathway called HLA class 2, which is mostly designed for exogenous antigens, very similar to what we had in class 1. Proteases are different, but small peptides will come and bind to a HLA class 2 and then come on the cell surface. And here, CD4 positive T cells can monitor what's going on HLA class 2. And again, if they find something which is not from our own body or genome, they can recognize it. So most of the cells that we have in the body, they have HLA class 1. But classically, we thought that most of HLA class 2 only present on antigen presenting cells. So why it's important in cancer? So as I said, almost all the proteins that we have inside the cell go through this antigen processing and presentation pathway. So if we have proteins that are unique to cancer and then go through this process and peptides from that proteins come to the cell surface, it means that we have some signals on the cell surface that immune system can recognize it. So then it means that in that case, we have some targets for cancer immunotherapy. So we have two general class of tumor antigens. It means peptides that are unique to HLA-bound peptides that are unique to tumors. One is tumor-associated antigens. They usually come from overexpressed proteins or cancer-testis antigens, or tumor-specific antigens that they derive from oncoviral antigens or neoantigens. It means peptides that sometimes we have mutations, and then that change in amino acid that happens because of mutation comes to the cell surface, and then we can see this neoantigen on the cell surface. So if a peptide comes from tumor-associated antigen or tumor-specific antigen, then this would be a target for cancer immunotherapy. So there are some interesting things about HLA. One is HLA is most polymorphic gene in the human body. So each of us has up to 6 different types of HLA class 1, 3A, 3B, and 3C. And what happens if we have an antigen like this? You see there's like a long protein. Based on the specificity that we have for each HLA type, only a small part of that antigen can bind to that HLA and present on the cell surface. So this means if we look at different patients with different type of HLA, from a known cancer antigen, we see different peptides on the cell surface. So it's somehow, you can't say personalized, but based on the HLA type, peptides that are present on the cell surface from one antigen is different. For example, if we have HLA A0201, most of the peptides that are present on this particular type of HLA have leucine and methionine position 2 and valine and leucine position 9. But if we go for HLA B5701, we have serine and threonine at position 2 and tryptophan and phenylalanine in position 9. So this is why we need to have a technology to understand from that particular antigen which peptide present on the cell surface, and it's why we need to have immunopeptidomics. So immunopeptidomics is the technology to find which peptide present on the cell surface. So we can start any type of material to start this process. Then what we usually do, we use a different method to homogenize this sample. It could be a biopsy, it could be a whole organ, it could be cell pellets. Then we generally use immunoprecipitation to pull down MHC peptide complexes, and if needed, we use high-performance liquid chromatography to reduce the complexity of sample, and then run it on mass spectrometer LC-mass, and then use some bioinformatics workflow to find the peptides, do some statistical analysis to estimate false discovery rate, and try to do some some quantification to compare the level of expression of particular peptides in different type of samples, and then several validation. So this is a very general workflow for immunopeptidomics, starting from any type of material to statistical analysis. So this method was not very popular maybe 10, 15 years ago, but now if you look at this paper published in 2022, you see so far around like 400 or 500 papers published that they used immunopeptidomics in their paper, and around 122 papers used immunopeptidomics for cancer. What's very interesting that now there are very big names in biotech and pharma companies that they have immunopeptidomics in their workflow, in their research. I mean, you see Roche is here, Genentech, and there are lots of small or like startups that are using immunopeptidomics in their pipeline to find targets for cancer immunotherapy. So it sounds that immunopeptidomics is straightforward, but what I want to show you here is that that's straightforward because it has its own complexity. Go to the next slide. Okay, so I tell you a little bit about one story that I worked for a long time. That's about proteasome splicing. So for many years, people thought that proteasome can only cut proteins and make peptides from a protein. But early 21st century, there was a few papers shows that proteasome not only cut proteins, but also it can paste two segments of a protein to each other and make a cis-spliced peptide. And then there was another paper which showed that it can do cutting for two different proteins and paste two segments from two different proteins, which is called trans-spliced peptide. So it makes the repertoire of MHC peptides or immunopeptidome much more complex. And this was very interesting and also challenging because we don't have any template for this cis and trans-spliced peptide in our proteome or in our genome. So normal workflows that we have in immunopeptidomics could not work well for them because in general proteomics and in general immunopeptidomics, we always have a template for our proteome, but we don't have this for spliced peptides. So it's why we had to develop a workflow, it's called hybrid finder, that we needed to do de novo sequencing to find candidates for cis-spliced and trans-spliced peptides. I don't go too much detail about how we do this, but we try to match our spectra to everything which are in the database. And if we didn't find any match, then we did de novo sequencing, find high confidence de novo sequences, and then use hybrid finder to see what's the source of this peptide, and then find peptides that cis and trans-spliced peptides. So we found these peptides, and we showed that a considerable proportion of peptides present by HLA class 1 across different HLA types are spliced peptides, cis or trans, and later on we found less proportion, but still a small proportion, like 2% of peptides that present by HLA class 2 are also spliced peptide. So it means if we don't use this kind of workflow and if we don't understand the biology behind generation of peptides on the cell surface, we miss peptides that are presenting on the cell surface. So why it's important that we don't miss them? So I just tell you this story about one of the very well-known cancer antigens, PML or GP100, which is a very well-known antigen in melanoma. So this is a sequence of this protein, and if you're on just normal immunopeptidomics, you expect to see 1 or 2 peptides or maximum 3 peptides from each segment, which was the case for us, that we found 3 peptides from this part of this antigen. But surprisingly, we saw more than 10 other peptides which coming from this segment, but in spliced form. So what happened? This segment somehow stuck in the proteasome, and then other amino acids like 1, 2, 3 amino acids from other parts of this protein just came and bind to the N-terminus of this small peptide here and generate new sequences that all of them was presenting on the cell surface. So we synthesized this peptide and confirmed that the sequence is right, and then we did some other studies for validation and see if they are immunogenic or not, which I'm not going to talk about them here. But ultimately, after all of this validation, now we have these peptides that you see here on the left-hand side in a clinical trial. that melanoma patients receive this peptide as a vaccine for melanoma immunotherapy. So this is how we develop a technology to find peptides that are presenting on the cell surface from a non-cancer antigen, and we are using them in clinical trial. So one other thing that I wanted to tell you clearly, that immunopeptidome is super dynamic. So everything that happens in the tumor microenvironment, treatments that we are doing could change the immunopeptidome. So this is a paper from Gabriel. So what he did, he treated triple-negative breast cancer cell lines with interferon gamma, a very well-known pro-inflammatory cytokine that we have in tumor microenvironment. And what he found for HLA class 1, after 48 hours treatment, we see around 3 times to 4 times induction in HLA class 1 expression. What was very interesting for HLA class 2, it was almost absent before interferon gamma treatment, but we see a considerable expression of HLA class 2 after interferon gamma treatment. So it's somehow we are mimicking what's happening in tumor microenvironment when we have a hot tumor, which means that we have interferon gamma there. Okay, so we see higher expression of HLA, but what does it mean in terms of the peptides presenting on the cell surface? So what you see here is the source proteins from known cancer-associated antigen or cancer testis antigens. And if we see peptides derived from this antigen in different situations, you see after we have interferon gamma, there are almost 50 cancer antigens, new antigens that we see peptides from them presenting on the cell surface. So we are expanding neoantigens or cancer antigens after treating with interferon gamma. And then when we look at the number of peptides, we see 4 times more peptide when we have interferon gamma. And very interestingly, 50% of peptides that derived from cancer antigen are only expressing by HLA class 2. The other interesting thing that we found was that most of the peptides that we see, which are in before or after interferon gamma treatment from cancer testis antigens, are shared with and without interferon gamma, what you see here in purple. There are a few peptides that only present in untreated condition, but there are lots of peptides that only present after interferon gamma treatment. So it's what we see in red. So it means this kind of treatment increases the number of targets that we can use for cancer immunotherapy. Okay, I just spent like a few minutes to talk about how we can use immunopeptidome for translational science. I had this slide before, but we have some challenges here. So the first challenge that we had was technique. In general immunopeptidomics, we need to have like big biopsies or billions of cells for immunopeptidomics. But now we develop a technique called peptide PCR, so we can use a small amount of material to have a targeted approach. So now we can use small biopsies and see which peptides are coming on biopsies. We don't need always go to cell culture. One problem was throughput. So the problem was that if for each sample, we needed to spend a few days to do immunopeptidomics, but now with new technique that we are using in the lab, so we are using Kingfisher and magnetic beads, and we developed this technique there, so we can analyze easily tens of samples per day. Then quantification was not perfect in immunopeptidomics, but now we are using some new techniques called data-independent acquisition, or TMT labeling, to do quantification as well as identification. And as I said, so there are lots of unknown sources for peptides present on the cell surface. So it's why we are developing several bioinformatics workflow to find new peptides on the cell surface. So we have this workflow. What we recently added to our workflow is that we are using different body fluids to see soluble HLA. So these are peptide HLA complexes comes from different organs to the different body fluids like blood or CSF to find them as a source for biomarkers. And we developed a technique, it's working well, so we can now find this as a new source for biomarker or antigen discovery. Okay, so I think I need to finish it. So these are the current projects that I have in we have in our lab. So we are working on different type of cancer, different type of pediatric and adult cancer. So you see some programs are quite advanced, like what we have in melanoma, breast cancer and ovarian cancer, and some of projects that we are in the very first steps. Okay, so hopefully I convinced you that immunopeptidomics is a powerful technology for antigen discovery in cancer. And now, after all these advancements that we have in technology, we can use it in translational or clinical setting. So cancer-specific modification on any level, it could be genome, transcriptome, proteome, peptidome, or post-translational modifications like phosphorylation. If they are unique to cancer, then we can think of using them as a target for cancer immunotherapy. The good thing for peptide HLA or HLA peptides is that they are on the cell surface. So the main challenge that we have in drug discovery that we cannot target intracellular proteins, we don't have this problem here. So if you have something in your mind that there is a particular mutation or gene fusion or RNA splicing or endogenous retrovirus that present in some particular type of cancer, that could be a target for T-cell therapy. The other thing is that we and others found that different treatments like radiotherapy, like using HDAC inhibitors, induce expression of particular genes which they are silent in normal situation. So again, peptides derived from that genes could be a target for cancer immunotherapy. Okay, so thank you very much. Thanks to all my collaborators and all funding bodies. Definitely thanks to my team. And we have open positions for postdocs and RA. So if you're interested to join us, please let me know. Thank you. I'm happy to take any question.
Terry, do you have slides as well? Do you want to do this now and we take questions at the end?
Yeah, we can do that as well. Yeah.
Yeah.
Can you guys? Oh, good. All right. Yep. All right. I'm just going to sort of talk about the phosphoproteomics, and then in the second half, I'll talk about the Olink technology. So why phosphoproteomics? So phosphorylation plays a sort of an important role in many cellular functions, including cell cycle, cell division, genes, transcription, motility, metabolism, even apoptosis as well. So you can think of phosphorylation as a like molecular switch that can turn on and off like a molecule. So therefore, it's very important to control the homeostasis of phosphorylation. So the balance of phosphorylation in the cell is very critical. And dysregulation of this balance usually can lead to disease, and it is often a driving factor in many cancers. So we can see here on the graph here, like let me just, how do I get there's a pointer. So yeah, you can see on the graph here, like there's in cancer, like dysregulation in kinases, which is the enzyme that that catalyzes the phosphorylation reaction. So dysregulation in kinases is more frequent in cancers compared to other diseases. So another reason why we want to use phosphoproteomics is that there's still a lot that still unknown about the phosphoproteome, and also the role of kinases inside the cell. To date, I think there's around 530 kinase, which is around 2% of the whole human genes, I think. But out of these 500 kinases, only around a third that we know the function of the kinases. So all the kinases that's highlighted here on that kinase tree in blue and red, or we actually still don't know the actual function of those kinases. So there's still a lot that's unknown. And up to date, there's around 200,000 phosphosites that has been reported, but only around like 7,000 or 8,000 of these phosphosites that we actually know the function. So there's still a lot to explore in terms of phosphoproteomics. And I think the more we explore, it will actually guide our decision to on how to develop more maybe precision therapy for cancer, for example. So how do we apply phosphoproteomics when it comes to precision medicine or precision oncology in that case? So generally, as I think Pouya mentioned earlier with the immunopeptidomics workflow, so the starting materials is usually very similar. So we'll start with either like a tumor biopsy or a patient-derived xenograft or PDX or even organoids. And in order to study phosphorylation by mass spec, what we have to do is to enrich for the phosphorylation. So we have to perform an enrichment, and there's several ways to actually enrich for phosphorylation. I'll come back to this bit a bit later. But after we do the enrichment, what we do is just we analyze the phosphoproteome by mass spec. And then if we have genomic data, for example, we have mutations, splice variant, or fusion, we can sort of integrate all these data with our mass spec data, and using the data and ultimately will sort of help us to stratify the patients, so that rather to use a standard treatment for for that cancer, for example, we can be more specific and more targeted in our approach, and like use treatment that will have the most impact on the patient's health. So as I mentioned earlier, so there's a few ways to actually enrich for the phosphoproteome, and if we interested in kinases more specifically, we can actually enrich for kinase before doing the before enriching for the phosphoproteome. And the most common way to like most people are using to enrich for the phosphoproteome nowadays is it's called metal cation enrichment, and one of the most common one is using titanium dioxide beads. So by using this method, so all this process, it's a very sort of manual process, and this limits us on how many samples we can process in one experiment. And usually, for most of our experiments, so we can process between 24 to 48 samples in one experiment, and it usually takes us between 3 to 4 hours per experiment. And one of our sort of issue with doing this kind of enrichment is the actual starting materials, which is around 500 micrograms. And this is sort of a problem when it comes to biopsies, which sometimes are very tiny, and we can't actually achieve that amount of protein lysate. So with all the like clinical sample nowadays, which usually comes like each experiment might come like with a few hundred sample, this sort of workflow start to become like a bottleneck. So with all this in mind, so we recently developed like a semi-automated workflow using the Kingfisher, so using magnetic beads on the Kingfisher platform that Pouya mentioned earlier, and using these magnetic beads from this company called Resin Bioscience. So what this workflow allows us to do is process up to 96 sample, and each experiment takes around 45 minutes compared to the 3 to 4 hours that the previous method took us. And also one of the major advantage of this workflow is the lower input requirements. So in that workflow, we actually require around 200 microgram of protein lysate. So how do this new workflow perform? So and in with this blue arrow here shows the all workflow, and then all the different column here are just different magnetic beads from that company using different buffer system. But comparing the magnetic beads with the all workflow, we can see we have like 1.2 to 2 times more phosphoside compared to the all workflow. And as I pointed out earlier, we're having more phosphoside with starting with sort of half the amount of starting materials, and which is a good thing when it comes to clinical sample, because we can start with lower starting materials and have close to double the amount of coverage in terms of phosphosides. So next, I just wanted to give you some example where we use phosphoproteomics in some cancer studies. So the first study is a sort of a pretty cool study where they studied how interaction between cell type are involved in prostate cancer, especially how interaction between the two different cell type can promote metastasis. And this was done all in vitro. So I won't go into much details on the method, but in a nutshell, each cell type has been stained with a different label and then mixed in a co-culture and then allow them to grow and interact together. But because they have a different label, we can tell which protein is coming from which cell type by mass spectrometry, and therefore can measure the different changes that are cell type specific, even if they are in co-culture. And for example here, mixing those two cell type here, we can see that we see an increase in phosphorylation of these proteins here by mixing those two cell type together, and then it like decrease in phosphorylation for those proteins here. And one of the analysis that we can do with mass spectrometry is looking at cellular processes. For example, which process is actually increased in which cell type, and and by doing this type of analysis, by integrating all these analysis together for this study, this led them to actually identify some top differentially regulated proteins and phosphosides, and one of them is actually TGM2 that play a role in the phosphorylation and migration in prostate cancer epithelial cells, and those sort of pathways or special or targets can be used to develop a therapy for precision medicine. So in this other example, which is sort of a more simpler experiment in terms of experimental design, this is an example where we can apply phosphoproteomics for stratification of cancer types. And in that case, it's breast cancer. In this study, what they showed is they identified this FGFR receptor, so which is a growth factor receptor, so the same family as EGFR. So looking at the phosphorylation status of these receptors, they could stratify between the different breast cancer subtypes. And more importantly, what we showed was high expression of FGFR4 in triple negative breast cancer, which is one of the most aggressive breast cancer subtype, was correlated with sensitivity towards one of the FGFR4 inhibitor, which is blue 9931. So these two are like a few examples where we can apply phosphoproteomics in our experiment. But some of our most common types of comparison would be like looking at wild type versus knockout, or expression sort of experiment, for example, expressing a kinase and then see how the phosphoproteome changes, or using a drug as well, and we can look how the phosphoproteome actually changes after using a drug, or between like control versus a infection sort of model as well, which is, I don't think it's as applicable in terms of cancer, but I think like there's like viruses like Hep B and C, which I think is which is linked to cancer, or like bacteria such as Helicobacter, which is linked to gastric cancer, for example. Those type of study, we can still do this type of study as well. So next, I'll spend maybe like the next 5 minutes to speak about the Olink. So before I'll sort of explain what is Olink, I'll sort of like say, why do we need Olink? So in general proteomics, and this slide actually takes plasma as an example. So for general proteomics, protein abundance is actually correlated with detection. So higher abundant proteins are more readily detected, or more easily detected compared to lower abundant protein. And usually below certain threshold, the mass spec start to struggle to actually detect this protein. And these are the proteins such as, for example, cytokines or chemokines. And that's where Olinks comes sort of to fill the gap. This Olink technology helps us to actually measure this low amount of protein. So how does this technology work? So the Olink technology works. So so the Olink is based on this technology called proximity extension assays, so short PEA. And how it works is they have two probes for each target that they want to measure. And by having two probes, it actually decreases cross-reactivity. And each probe actually has a DNA tag. And the DNA tag will only be able to be amplified by PCR when they are in close proximity. So in this workflow, there's actually a pre-amplification by PCR, but the actual quantification is actually done by qPCR. So I'll just try to play, if I can, this video, which actually summarizes that Olink technology. whoops. So yeah, so this is sort of an overview how Olink works. How do I go? Next slide. Can we move them? Oh. So so on that slide sort of show like the different panel that we have for Olink. So starting from the left here, it's called the Explore panel. And these are usually for large discovery studies where I think this is an old slide. Like currently we can measure up to 3,000 protein, but Olink is actually working towards a new panel that we can actually measure up to 5,000 different protein from this Explore panel. And there's also more targeted panel called Target 96 or Target 48. And these are pre-designed panel. So we have panel, for example, for oncology or immuno-oncology, cardiovascular or inflammation as well. And we even have a panel for mouse. And then the last panel is called Flex or Focus panel, which are sort of custom-made panel. So you can sort of pick and choose which protein biomarker you want to actually measure in your sample. So at Monash here, so we mostly focus to do this Target 96, Target 48, the Focus and Flex panel. And if you want to do the Explore panel, we work in collaboration with WeHi, where they actually have the NGS equipment to actually run these sort of panels there. So, although Olink sort of has been designed primarily to study plasma or serum sample, this technology can be actually extended to study other sample type, like cell lysate or tissue lysate, which I think most of us here will be working with. But yeah, since the Olink platform has been designed for serum and plasma sample using these other sample types, it's usually requires a little bit of tweaking, but it can actually be done on this platform. And I don't have any summary slides, but we are happy to take any questions, I think now.
Thank you very much, Terry and Pouya. This is really exciting to have this Olink technology available to us at Monash. We have time for a few questions. If you'd like to ask a question, please unmute and turn on your video, so if you'd like, so we can see you. I might start with a question while others think about it. I was wondering about the starting material, and are there any organ or tissue types that are really challenging for proteomics applications? can you comment on that?
So if you're asking about immunopeptidomics and proteomics, yeah, there are some challenges that we have, for example, for immunopeptidomics. Sometimes we don't have any HLA expression on some particular type of tissues, and I mean, this is one of the mechanisms of immune evasion and in cancer. Usually for us, it's a little bit challenging to work with brain samples because we don't have enough expression of HLA there as well. When I say challenging, it's still possible. It doesn't mean it's impossible, but we always need to optimize methods based on if it's like a new tissue or sample that we didn't work before that. We need to optimize some process that we have.
Okay, so there isn't any cancer, like you know, sort of if you're working on this type of cancer, don't even think about proteomics. It's not going to work. There's no such.
No.
You can always try and optimize your assay.
Yeah. I mean, in terms of So for example, if we are working on general models like human samples or like different animals that we have the proteome map for them or genome map for them, that's always possible. Sometimes we don't have enough genomics data to know what proteins is in a particular thing. So that would be challenging.
Do we have any questions from attendees? So when you say genomics data, do you need whole genome data, or can you work with?
Usually it's exome. It's exome data.
Right.
I mean, having said that, when you are looking for non-canonical MHC peptides, for example, yes, there we need also introns and the whole genome.
I can't see any raised hands. Well, I have a lot of questions, Pouya and Thierry, but I might just go find you, and I encourage everyone who's here and thinking about using any of the approaches that Pouya or Thierry have talked about to just contact them. So I think you are always open for collaboration. So I know these guys, and I know they're always happy to help and collaborate. So yeah, if you're thinking about this, applying any of this in your research projects, I'm sure they would like to. They'll be happy to talk to you. So, all right, because we're coming close to 3, I would just, if there aren't any other questions. I just want to thank Pouya and Thierry again. Thank you everyone for joining, and we look forward to seeing you at our